The overall goal of this procedure is to measure the kinetics of nucleotide binding and dissociation during the ATP turnover cycle of a kinesin. This is accomplished by first preparing the kinesin protein either free of bound nucleotide or loaded with fluorescently labeled nucleotide. Next, the prepared kinesin is mixed with nucleotide using a stopped flow fluorimeter, and the change in fluorescence associated with binding or dissociation of fluorescently labeled nucleotide is observed. Then, the change in fluorescence is fit to an exponential function to obtain a rate constant. Finally, the measured rate constants are deconvolved, and rate constants are attributed to particular transitions of the ATP turnover cycle. Ultimately, the combination of fluorescently labeled nucleotide and stopped flow fluorescence is used to measure the kinetics of nucleotide binding and dissociation for a kinesin.
Demonstrating the procedure will be Jennifer Patel, a postdoc, and Hannah Belsham, a grad student from my lab.
After determining the fluorescence intensity of kinesin upon binding of MANT-labeled nucleotide as outlined in the text protocol to a solution of up to 20 micromolar kinesin in magnesium-free buffer, add 1 millimolar of EDTA and 1 millimolar of GTT. Incubate the solution for 15 minutes at 25 degrees Celsius. Set up a gravity flow G25 Cephadex gel filtration column and equilibrate with at least 3 column volumes of a magnesium-free buffer. Then, load the kinesin solution into the column to separate free nucleotides and EDTA from the kinesin. Prepare